So today we have Karunakar, who is an associate professor at the School of Life Sciences at JNU. He leads the biophysical and biomaterial research laboratory, where his work primarily focuses on the interaction of biophysics, biochemistry, and nanotechnology. His research interests center on understanding the.
Thank you. So my name is Karunakar and I want a very quick introduction. Just what you want to be. We can go through. Name and what is your age? Okay, settle me. Scientists who had three researchers, PhD students. And they went to a planet outside. And they were exploring. They had a space station there. Not space station. They had a station on the planet itself. And the scientist asked the students, please explore whether there is a moon or not in the sky. Got it? So first student went and he looked at the window, tried to explore as much as possible, and he makes his report. There is no moon in the sky. The second student went, he tried to peek out from the window a little bit more. He became courageous and with the trace he came out of that house, tried to explore all the corners, and he went to the roof and tried to see as much as possible. Then he comes back and says there is no moon in the sky. The third student went outside. He did the same thing the other student did, but he then said, I am not happy with what I explored. Give me some time. Then he repeats that experiment. That means going and looking at things next day, and one day he saw a crest. So next day he saw a crest. Crest means you understand, right? So Amabas ke din, we don't see moon. So if somebody from outside comes on earth on the day of new moon day, he will not see the moon. And he comes back to his planet. So he will never say that there is a moon. Got it? So in research, it is very important to realize this. If you see something, doesn't mean that that's, if you don't see something, that is also some hidden knowledge inside that observation. So research is to explore the failures. If you do an experiment and you don't find any result, you should not be dragged by that result. हुआ नहीं, inhibition नहीं हुआ या कुछ चीज जो आप चाहते थे, that doesn't happen. So that is a very important quality that you should inculcate in your wherever do whatever research profession, research field you do, but this is very important. So never be frustrated when you don't see any results, or if you don't qualify any, even for research entrance exam or interviews. So don't be frustrated. So be courageous, and that is a very important property for research. So coming back to the topic here. So I am going to introduce you to amyloid aggregation. How many of you have understood or heard about amyloid aggregation or amyloid diseases? How many? Amyloid, Tom? No one? Alzheimer's disease, Parkinson's disease, have you heard about? But you have never heard about amyloid. Okay. So we'll get to that. Amyloid aggregation is a aggregation. Protein aggregation, have you heard? So amyloid aggregation, as the name indicates, is one kind of a special or specific type of aggregation of proteins. What is that amyloid aggregation? And then how to target that process. These are the two things we'll go through when I'll be taking you through the slides. Pointer नहीं है? Pointer है क्या? ठीक है. If I were to ask you what is the definition of life in a scientific term, everybody knows it's the DNA. DNA defines life. Okay. And cells are the functional unit of life, be it unicellular or be it multicellular. But DNA has the information only for proteins. If you look at the DNA codes, it's genetic code, 3-letter code for amino acid sequence. So DNA doesn't do anything else, but just specifies a sequence of a protein. Got it? So in other words, if you can extend that statement, if DNA defines life, proteins in fact drive life, because DNA only codes for the proteins, and once these proteins are synthesized, they are directly or indirectly involved with almost all the metabolic processes that is required for manifestation of the cells, tissues, and organisms. So if proteins are very important workhorses or macromolecules for the life or for life metabolism, then we understand proteins, and we have certainly developed a lot to understand protein structure, function. Every disease is directly or indirectly associated with a protein. So I don't have to introduce what is protein here. But left side is kind of a protein, insulin, and this is the structure of that. Now, if you have a protein in your system, like a cell or cellular environment, that protein is a folded structure. So there is a process called protein folding. So the DNA, it is better if you have a laser beam, but anyway, you just look at that. Ribosome synthesize the polypeptide chain, and the polypeptide chain has the sequence as per the DNA, and the polypeptide chain then folds itself into a three-dimensional structure. Now, this structure is very important, which is given as native structure, very important for the function for which it was synthesized. Now, this process of protein folding is very fundamental to biological systems. However, there are many factors which can influence this process, like pH, temperature, deletion, mutation, so many factors. And if these factors influence this process, then there is a possibility of misfolded structures, or protein not going into its native functional state but going and arriving at a misfolded structure. And misfolded structures are aggregation prone. That means they have a tendency or intrinsic property to self-assemble into higher order structure. And if that process is triggered, then it is energetically driven. That means proteins will be trapped into an aggregation pathway. And that is the process of aggregation. Now, in amyloid aggregation, which we are going to discuss today, the higher order structures, so protein is aggregating into a higher order structure, have a typical signature, structural signature, that is cross beta. It is schematically shown here. Beta strands, beta strands, beta strands. So 2, 3 protein self-assemble by maintaining a beta strand. It is called cross beta because the axis of the beta strand and the axis of the fibril cross each other. Because this is where the fibril axis grows, and these are the strands. So the definition of amyloid fibril formation, if we can mention it, aggregation of proteins and peptides into a higher order fibrillar architecture, maintaining a cross beta conformation. Now, this process is a conformational switch. Let me say phase change, soluble to liquid to solid, phase change, and it's a conformational switch. Whatever the secondary structure composition of the protein, end of the amyloid fibril formation, it will be rich in beta strands. So it is mostly alpha to beta transition. So it's not only a phase change, it's also a conformational switch from native to a beta seed rich higher order fibrillar structure. So why would it be a problem for the body system? Why would it be a problem and lead to disease? Because these fibers, amyloid fibers, which are rich in beta strands, are cytotoxic. They can kill cells, they can cause necrosis, apoptosis mediated cell death, they can cause membrane damage, mitochondrial damage, lysosomal damage. So many damaging effects are directly linked to amyloid aggregation of proteins and peptides. And because of this reason, amyloid aggregation of proteins are linked to diseases. So, if you see the history, the discovery of amyloid diseases, Tell me any amyloid disease. We just discussed Parkinson's and other Alzheimer's. So 1906, remember 1906, Alois Alzheimer, the doctor was treating a patient. Her name was Augusta, and she was suffering from a disease which was very new kind of symptom that time, memory loss. Memory loss was a very new symptom that time. Now it is very common, right? What was your launch in 2 days back? 3 days back? What was your launch 3 days back? And what a difference in memory. So nowadays dementia is a very common symptom, right? When movies are being made on dementia. So that time it was very new kind of symptom, memory loss, and she died at the age of 55. The severity, the disease severity became more and more complicated. So the doctor wanted to see what is actually, and the brain tissues he examined the patient, and he found black dots. I could not bring that, but this is if you see the top picture, that is the Alzheimer plaques of the tissue. I think यहाँ पे भी आप printer ले सकते हैं, right? If you see here, those are the brain tissues cross section of Alzheimer patient. So these black dots, and it was first discovered by Alois Alzheimer. Remember, 2026 we are talking now about Alzheimer's disease, and 120 years have passed. We have certainly progressed a lot in understanding cell biology, improving our knowledge on molecular biology, genetic engineering, tissue engineering. We are making artificial organs, computational biology at the top. However, we have not yet understood. Definitely, we have improved our knowledge in understanding the mechanism of amyloid aggregation, but we have not yet been able to find out a strategy to target these diseases. That is why you see Parkinson's disease patients, Alzheimer's disease patients, still they are suffering. There is no cure at all. They have to suffer. So, amyloid disease has expanded from Alzheimer to so many other diseases. There are 55 diseases right now which are directly linked to amyloid aggregation. Question? If you have any question, anything related to this or any, just feel free to ask me. Do you know what is prion? Anyone? Do you know what is prion? Proteinaceous. So prion is the Nobel Prize. It got Nobel Prize, no? So generally, the diseases are caused by infection, by bacterium, virus. No, prions are protein particles. They are proteins. Misfolding is every protein can misfold, but they are proteins, and the prions are a normal protein can misfold or form some attain some other conformation, which becomes aggregation prone. What I told in the previous slide. If you have a normal protein, no issue. If during the protein folding or due to mutation, many factors, if it forms a misfolded structure which becomes aggregation prone, then that is going to self-assemble, mean form aggregation. But if it interacts with normal proteins, again, a misfolded protein interacts with normal protein, then that interaction will change the conformation of the normal protein towards misfolded structure. That is why this misfolded proteinaceous structure has the ability to change the normal proteins into prion structures. So it acts like an infectious proteinaceous particle. But remember, 20 years, I think he got it in 2000, Kushner, no? And right now, that concept is very much diluted, and many other structures can also influence aggregation of amyloid, aggregation of other proteins. So maybe we will have more information. But that is why prion, what is the question? So prion is forming amyloid structures. So prion disease is also there, mad cow disease. So resultant of all the diseases, tissue deposits, and tissue deposits are like these plaques, black dots, inclusions, plaques in Alzheimer's disease, Lewy bodies in Parkinson's disease. So now this is something which is a process of amyloidogenesis. It starts with a peptide. If it is A beta peptide, then Alzheimer's disease. If it is alpha synuclein, then it is Parkinson's disease. If it is prion peptide, then prion disease. So many diseases. घड़ी नहीं यहाँ पे. So coming back to this slide, just to revamp, at least you have understood now normal proteins can be misfolded due to many factors like pH, temperature, degradation, mutation, maybe cumulative factors, and they become misfolded structure. Then this guy will become a misfold, undergo a process called amyloid aggregation, and this is the fibular structures. And if you look at the structural properties or characteristics of these fibers, then they are actually maintaining a cross beta conformation. Beta strands, beta strands, beta strands in the fibril axis. That is why it's called cross beta structure. So definition of amyloid fibrils. Now let us see how to target this. In fact, these are some of the amyloidogenic proteins for your information. This is A beta, and this is the structure of A beta. So people have solved, because we are now cryo-EM and X-ray structures are possible. So people have solved the amyloid fibril structures of many proteins, and this is the amyloid fiber, and this is actually how does it look. So there are so many some examples I have given here. Look at, if you go to the right kernel, you see strategy 1, then strategy 2, and strategy 1 and 2. We'll get to that in the next slide. As I mentioned, you have a DNA. DNA is expressing the protein, and this protein is folding itself into the native structure. It does its function, no issue. But if gets unfolded, misfolded, many factors, formation of misfolded structure, it will start aggregating and it will form amyloid structures, and these amyloid structures are ultimately going to form the plaques in the neuronal cortex or the brain tissues, and then they are going to have their effect, Parkinson's disease or memory loss, whatever, depending on the part of the brain where they are getting deposited. Now one strategy, number one, is block the gene. If there is a protein which amyloid aggregation leads to disease, and you block it, you don't have that protein, no disease. But remember, every protein has a function, very essential for the cell. So there is no protein which is useless. So if you block that protein, maybe you will be losing some of the important functions. So it is a strategy which is definitely going to have some side effects. Second attempt, which is most of the amyloid researchers are focusing on, is trying to inhibit the initiation of amyloid aggregation. If you could prevent the process of amyloid aggregation from the beginning, that would be of great help. And third one, which is also very important, is some strategy to dissolve the plaques. Like you are having a formulation or therapeutics, and you add to the plaques or higher order structures or amyloid fibers, then it starts disintegrating the structures into non-toxic soluble species. So number 2 and number 3 are very important, and people are trying to develop therapeutics, and one of the approaches is nano strategy. At least you have understood this slide. As I mentioned, any question, anytime, you can ask me. Our lab is working on some specific amyloidogenic proteins. I cannot share it in detail, but some of the proteins are amyloid linked diseases like Alzheimer's disease, insulin, alpha synuclein, and we are trying to understand the mechanism of amyloid aggregation, because more you understand the mechanism, better you have a strategy to target that process. Now, as I mentioned here, just remember the problem is initiation of amyloid aggregation. A protein is very much functional and native structure. What happens to that protein that it becomes aggregation protein? This is an important question. Why would a native structure attain a structure misfolded structure? Even though it attains, why would that misfolded structure undergo an aggregation process? These are the questions. More you know about these processes, better would be your strategy to target, because that is initiating that process in the beginning. So we are trying to do that, and the same time we are trying to formulate something, and we are now using nano formulations to dissolve the plaques. You make the amyloid aggregate and you add some therapeutics, nano formulation, natural compound, or a combination, so that the amyloid aggregates are getting disintegrated into non-toxic soluble species. Remember, we have to get rid of toxicity. So it's the toxicity of amyloid structures which is causing the disease. So if you are disintegrating amyloid fibers, but the toxicity is still there, then it won't help you. Got it? Now, why nano? Just 2-3 minutes. All of you know about nano. Why nanoscience is very much at the frontier research domain, irrespective of all branches of science. Anyone? Nanoscience. You heard about nano, right? Nanotechnology, nanoscience, nanoformulation, nanoparticles. थोड़ा थोड़ा interactive हो. What is nano science? ठीक है. Good answer. Any other answer? Why nano? Even now, all universities have a nanoscience department. Oh. This is my understanding. You know about it, but let me explain what I believe, or I think it's nanoscience is nothing like out of blue or something different branch of science. It's a part of our science, but looking at, as he correctly said, looking at a different dimension. And if you look at this picture, I have taken it from White and White. All of you heard about biochemistry book. Now left side is protists, the most primitive organism. Got it? Right side is human beings, us. And the middle is Arabidopsis, that's the plant species. Now if you take Tata binding protein, transcription factor, right? And the structure of Tata binding protein is solved for these three species. And it is given there. It's given in the right dimension. So protists and human being, if you compare the size and compare the protein structure, so it has also a Tata binding protein, that is also, we also have Tata binding protein. The size of the organism, huge difference. And even these organisms are apart from each other by billion years of evolution years, right, of time. But still, they are maintaining the basic macromolecules like the proteins, this is example, at a unique or defined dimension. For example, hemoglobin of a rat, hemoglobin of an elephant. This simple question I can ask in another way. Is the size different? Animal size is different. Hemoglobin different. Why? If you can give some inputs, why? That's talking about genetics. I am talking about the concept of nano. What you said is correct. ये माउस का छोटा छोटा हाथ होता है, हमारा बड़ा बड़ा है, एलिफेंट का, जिराफ का इतना बड़ा है. तो टिश्यू वाइज वी आर डिफरेंट. But if you go to the size of the cell, if you go to the size of the macromolecules, proteins, it's the same. Why? So this is a kind of a question which I want you to think about, because if you more think about it, you will answer. So this is, I am giving this example to get to know about the relevance of nano. So biology operates at nanoscale. This is what we understand. If we can design at the nano dimension, we may be able to better intervene biological systems. If my hemoglobin is 100 times larger than the hemoglobin of a rat, because the size is, let's say, 100 times, when my hemoglobin cannot travel, अगर hemoglobin नीचे चला गया, ऊपर नहीं आ पाएगा, because we are governed by physico-chemical forces. Earth की geological Earth is actually controlled by gravitational force. Got it? So if hemoglobin is heavier, ट्रांसपोर्ट नहीं हो पाएगा, प्रोटीन बड़े बड़े हो जाएंगे, कैसे होगा फिर? So biology needs to accommodate itself according to the physico-chemical forces that it is governed by. Maybe when there is another planet where the gravitational force is 10 times higher, maybe the animals will have bigger proteins. Maybe. Got it? So because of that reason, the nature is very smart. He maintains the biological macromolecules at a particular dimension. What you said is 1 micro to, is like 1 nanometer to 200 nanometer. In that range, you will see all the macromolecules. Antibodies are like 300, 400. So more we focus that dimension and we design our therapeutics at nano dimension, better we may be able to better intervene the biological events. And one of the biological events that we are discussing today is amyloid aggregation, and that is also a nanofiber. In a material science, amyloid fibers are defined as a nanofiber because they are maintaining a 10 nanometer width. Amyloid fibers. So second part is how to target amyloid aggregation. At least every disease has a molecular origin, and for amyloid linked diseases, this we have been discussing since 10 minutes, 15 minutes. It is that conformational alteration that converts soluble native structures into aggregation prone species. A soluble native functional structure is converted into an aggregation prone structure, and that is a molecular event, and that is beginning the amyloid aggregation. It is beginning aggregation process leading to formation of toxic fibers, leading to apoptosis, cell death, neurodegeneration, and the disease onset. So how to target that? Getting some, like it's a big, it would be a big information if we really understand this, because amyloid aggregation, understanding this would help us in understanding the mechanism of amyloid aggregation. Let's see. This is the structure of a protein. Fundamentally, this is the structure of a protein, and this protein aggregates under certain conditions. As I mentioned, there are many factors which can trigger aggregation. If you have a protein at pH 7, for example, it does its function, no issue, it's soluble. But if you change the pH, add some additive, or take a mutant, or increase the temperature a little bit, 40-45 degrees, then you start see aggregation. And this aggregation, if you look at microscope, suppose is from amyloid fibers. That means that process is initiated in the same protein. So to understand that, we have to go to the MD simulation to see what is happening in the molecule. So this is a protein which forms amyloid aggregation in a condition. And this, if you look at protein structures, if you have Primal Discovery Channel, you have looked at some structures. Have you looked at structures in some of the platforms? Anyone? Have you seen protein structures in the books or in the publications? So those structures are static, is not it? जैसे कोई बोलेगा कि आपने करुणाकर सर का फोटो देखा है, फोटो स्टैटिक है, अभी के मैं स्टैटिक नहीं हूँ ना. There are vibration, I am dynamic. Even I am standing still, lot of vibrations. It's dynamic structure. So dynamism is fundamental to life. Remember that. Even though it is very still, but still the electrons are vibrating and revolving. Is not it? So the dynamism is also central to the protein structure. Try to understand this. So this dynamism is an intrinsic property. So even though we say protein is maintaining its native structure, but it is still in a dynamic structure. And that dynamism if I can little simplify this sentence, the dynamism is fluctuation. The protein is fluctuating, but the fluctuation is within the threshold that the active site is not disturbed. Still, it can do its function. And we know protein structures are stabilized by pH, hydrogen bond, non-covalent interactions, hydrophobic. You know that biochemistry, how the protein structure is stabilized. Polypeptide chain folds, alpha helix fluctuations. So there are so many intermediate structures are formed. And sometimes the alpha helix is broken, sometimes the loop structure is formed. And this is happening in the real time. That means when the protein is exposed to a condition, and we know that condition triggers protein aggregation, this is happening at the molecular level. The structure or the fluctuations are so that some of these intermediate structures are aggregation proof. If it loses its secondary structure, tertiary structures, and exposes its stretches of intrinsic disordered polypeptide chain, then these chains with hydrophobic residues will try to interact with similar species. So, in a very broader fundamental understanding, if you have a native structure, the hydrophobic residues are in the core of the structure, because globular proteins have hydrophobic residues located in the center, and it is a globular protein. If you increase the temperature, mutation, any factor which misfolds it, then misfolding will expose the hydrophobic groups or disordered zones. Same thing. So similar structures with exposed hydrophobic groups will never stay in isolated condition, because hydrophobic groups always try to interact with another hydrophobic group so that they can expel water. They will interact. So hydrophobic interaction will be there, but now two proteins are interacting. So that is actually initiating self-assembly. So this is a very initial event that begins aggregation of any protein, be it insulin, A beta, alpha synuclein. That is why there is a conformational switch from alpha to beta in general. Anyway, I'll skip some slides because of time limit. Time कितना हो गया? So this is A beta. I'll just go through the slide, and this is the fiber of A beta, and these two arrows are there. And as we understand now, this looks like a three-step process, but it is very complicated and very complex. So many structures are formed, so oligomerization. Even though after formation of higher order structure fibers, these are getting deposited in the tissue. And the prion concept, I'll come to that. So because we have understood that hydrophobic residues getting exposed in the misfolded structure is one of the initial events that triggers aggregation. So we wanted to test this hypothesis. If you have a protein exposed hydrophobic group, and then we add a nanoparticle which is coated with hydrophobic group, then it will try to interact with that, and if it interacts with that in an efficient way, then it may block the protein-protein interaction. And if you block the protein-protein interaction, you are actually indirectly blocking the aggregation process. That was the hypothesis we thought. The first nanoparticle that we synthesized in our lab is with this concept. So we made gold nanoparticles, silver nanoparticles coated with aromatic amino acids. They are the same amino acids. So if a protein has a hydrophobic group, it's the same hydrophobic group which is coated with the nanoparticles. And proteins, as I mentioned, proteins are the nanostructures. So nanoparticles are also the same dimension. So they can interact. And this is just one example. And these are the nanoparticles, the electron microscopy. They are homogeneous and spherical. And these nanoparticles, when you add to an aggregating protein, then they are inhibiting. This is one of the example. If you see left side, the red curve is the aggregation of the protein. And then in the presence of different doses of nanoparticles, that process is suppressed. You can see that. And it's a dose dependent inhibition. Now this same thing for that. Important observation is, which I want to highlight here, that if you add only hydrophobic amino acids, these are tyrosine coated nanoparticle, tryptophan coated nanoparticle, and phenylalanine coated nanoparticle. But if you add only phenylalanine, Only tyrosine. They are also hydrophobic, but you don't see that inhibition. That means it's the nano which is interfering with the proteins in an efficient way. So that is why the relevance of nanoformulation. The efficacy, the bioavailability, and the functional properties of the compounds which has some anti-amyloid activity. Suppose you have a natural compound which is anti-amyloid. If you make a nanoformulation of that natural compound, the efficacy, bioavailability will greatly enhance. This is what we have been working on, and the course of 2, 3, 4, 5 years, we have synthesized so many nanoparticles. Can you explain me why this is happening? That single hydrophobic group is not preventing, but the same hydrophobic group, when it is coated on the nanoparticle, is preventing. Why? Let's say it is interacting. It's interacting, that's why preventing. Why would it interact? Any physics guy or any? आप थोड़ा आंसर नहीं दोगे तो मुझे लगेगा आई एम टीचिंग समथिंग विच इज गोइंग. बोल सकते हैं. So you remember this question, ask me when we end this. Last में discussion करेंगे. So these are some other nanoparticles. This is proline coated, and when you have nanoparticle, you have to characterize nanoparticle. You have to do the biocompatibility, stability. Particles should be stable when you go to the blood solution, pH change. So a lot of things to be. It's not just making the nanoparticle and done. So that's what we do. Once we finalize that or establish that bioavailability, pH stability, thermostability, and the nanoparticles are not aggregating, then we go to the anti-amyloid studies. And once we do anti-amyloid study in vitro, so you study aggregation of protein in the presence and absence of nanoparticle, and you get an idea this is in vitro. But what is more important is to get into a therapeutics. Like you have to go to in vivo system. So if a nanoparticle is preventing aggregation of A beta, it's not sure that it is going to prevent Alzheimer's disease, because Alzheimer's disease is caused by A beta aggregation, but the actual cause is tissue deposition at the neuronal cortex. If you remember the slide, so left side, try to understand this is the signal which gives the signal for aggregated structure. The black one, where the cursor is. So you have a aggregated structure of amyloid fiber, and you take a reading. TST is a amyloid sensing dye. You get that reading, and if you don't see a change here, it's constant. So amyloid aggregates are very stable. They don't go back to solution. So you have that suspension, but the moment you add these nanoparticles, and there is a decrease. In the previous slides, I showed it is inhibiting. So a monomer is aggregating, it is preventing aggregation process. And in this exploration, we show that even though you have already amyloid fibers are formed, if you add these nanoparticles, it is reducing the amyloid structures into soluble species. We took that sample, the disassembled sample, and look at the monomer, and we saw that monomers are coming back from the fibers. So this is certainly, this gave us a very good direction to clearly go into in vivo mode. So if go back to this, inhibition of aggregation number 2 and promote disassembly of amyloid. So these nanoparticles are inhibiting protein aggregation. They are also disassembling amyloid fibers. So we have done some explored lot of natural compounds, and these are the natural compounds you have been eating every day. Everybody knows Indian curry, like spicy, capsaicin, piperine, and all these compounds are there. And remember, when you eat Indian food, it's not only the molecule that it's a formulation, because extract, then milk, it's a formulation. In the formulation, they also form some nano emulsions. Like turmeric is not taken as such in water; it is taken in milk. Can anybody tell why? Turmeric has a compound curcumin. Curcumin is taken in milk. Why? इसमें भी एक नानो छुपा है. Can anyone tell? Everybody knows about that, ना? अच्छा काम भी करता है. क्यों? क्वेश्चन को लिख कर वो लास्ट में डिस्कशन करेंगे. I'll just go through the slide. Just to say that these are the structures of one, and if you see the light side, the C is the larger magnified view, and they are very uniform. If you try to focus on the surface of these nanoparticles, you see a corona like a line, and those are the molecules which are coated. And this is 5 nanometer, below 5 nanometer, and which is very good for blood-brain barrier, crossing blood-brain barrier, because amyloids are formed in the brain. So if you are developing a drug, it has to cross blood-brain barrier. So 2 nanometer, 3 nanometer structure nanoparticles are the best candidates. And we have done a lot of work on this piperine coated gold nanoparticle. It is now in the RAD model, and we are so hopeful that we will modify this and we'll get into dissolving and treating amyloid disease. I don't know, sometime soon. I asked a question why nanoparticle is working, right? We'll explain this. So every protein has an aggregation prone zone. Left side is a protein structure which is predicted aggregation prone region. The red zones are aggregation prone. And the nanoparticle, the hydrophobic residue or the natural compound which is inhibiting is actually binding to that aggregation prone stretches. Okay. Now that is good. If you are having a natural compound which binds to the aggregation prone of the protein, then it is going to prevent its aggregation. Now, if you look at the chemistry, the dynamic protein ligand interaction, it's dynamic. They are binding, and then equilibrium is there. The binding affinity is also there. The binding affinity, and then two molecules bind in a system by collision. You know collision theory in chemistry. Two molecules has to collide, and collision happens when you have a Brownian motion. Molecule has to move around, right? So these lot of factors are there. So small molecule and the big protein, and the amount, maybe it needs 100 collisions to get to that binding site. And if you have, because the small molecules have high Brownian motion, they will vibrate in a larger frequency. So if you make a nano formulation, then it is same dimension as the protein. The vibration of the protein structure in the solution, Brownian motion, and the Brownian motion of the nanoparticle is the same range. So if they are coming like this, then there will be a sustained interaction. The molecule has to heat, and the heat should have enough time for the interaction. Second thing is the constraint. अब अगर molecule को आप hold करके interact करवाते हो, direct interact करते हो, so you are holding the molecule through the nanoparticle surface. So the molecule has a constraint. It has a structural constraint. So after binding, it retains its structural constraint. So the binding is facilitated. That is why nanoparticles, if they are coated with some compounds or ligands, they become more efficient in binding to the protein. So this is one of the reasons. I just want to show. So Parkinson's disease is caused by feeding paraquat. If you take a fly model and if you feed paraquat, it will develop Parkinson's disease symptoms, and that is one of the best model for studying Parkinson's disease model. So this is the fly, and the control is this is the brain, sectioning of the brain, and the control is faded green. So that means the brain is normal, and you gave the paraquat, and the bright spots, the green spots, indicate the apoptosis in the brain. So it is causing neuronal damage. There are other studies which are highlighted in that paper. So this fly is fed with piperine coated gold nanoparticle, because we had all prior studies, and this fly is fed with piperine coated gold nanoparticle after giving paraquat. That means pre-treatment, post-treatment, and you see it is completely vanished. So it is the nanoparticles that we have examined in the in vitro system is actually working in an in vivo system. It is preventing PD symptom, and it is also reversing the developed PD symptoms. So that is we are so much into this in vivo model, and hopefully in 1 or 2 years we will establish this in mouse model, and we are trying a combination so that this efficacy can be enhanced with less with more biocompatibility. Then we are talking about nanoparticle. This The left side image is actual simulation of a nanoparticle computer simulation. It's not schematic diagram. So the nanoparticles look like this if you really want to see. And this is just 2-second video because bada video. So this is the amyloid structure and this is the nanoparticles. There is a possible of interaction. They are interacting, and this interaction is somehow disrupting the amyloid structures, and this interaction with the monomer is protecting the monomers to going into amyloid aggregation. Both way it is working. So hopefully this will lead to a strategic development of therapeutics. Another 5 minutes is okay or ho gaya? This is little bit one step ahead because I want to mention this. It's not that simple as we have discussed. Amyloid aggregation prevention. The tissue deposits that we are the patients having, Alzheimer's disease, let's say, and we know Alzheimer's disease caused by amyloid aggregation of A beta. So you get A beta peptide in the test tube, start initiating aggregation, you'll see fibers. But in the body, when A beta is aggregating, it's not aggregating in an isolated environment like in the ependrofe. So this is a picture I have taken again from Voigt and Voigt, and this is the question I want to ask is, if a protein is aggregating and it's aggregating in an environment where so many other molecules are present, other proteins are also present, how does the aggregation process of one type of protein affect the aggregation propensity of other proteins, particularly the proteins remain in its vicinity? It's a very important question, and there lies the prion concept. So if you have a protein which is misfolded and it encounters a normal protein, then what would be its effect? And that answer is very, very scary, because if you have a aggregated structure which is going into a pool of normal proteins, then that aggregated structure can trigger the aggregation of other proteins. Story doesn't end here. So aggregation of other proteins will also lead to formation of amyloid fibers, and we know amyloid fibers are cytotoxic entity structures. So that is why prion concept is expanded now, because prion concept was based on only that prion sequence. But now people are saying, and in our lab also doing research in that research, any protein which is undergoing amyloid aggregate, take that amyloid aggregate of a particular protein and he add it to other protein monomers, A, B, insulin, lysozyme, beta, whatever the protein, it will trigger aggregation of other proteins. The specificity is not there. The reason is the cross beta structures can trap the proteins by finding suitable functional groups, and once they trap it, then they will change the conformation of the normal protein into an aggregation prone structure and trigger their aggregation. This process is called cross seeding. At least remember this, cross seeding. And two monomers can also come together. Protein A, protein B, they can come together. That is called co-aggregation. And these two events are also fundamental to amyloid aggregation. So when we try to understand amyloid aggregation, try to understand the mechanism of amyloid aggregation, we have to also understand co-aggregation and cross-seeding. At least this much you remember. And we have been working on that. If you see left side is insulin intermediate, insulin native, and insulin intermediate. So these proteins, intermediate structures are interacting with each other. Now talking about hetero, like left side is native structure of insulin, native structure of lysozyme, native structure of beta lactose, three globular proteins. They are interacting. This is kind of a simulated study. Right side is intermediate structure of beta lactoglobulin, intermediate structure of insulin, intermediate structure of lysozyme. And if you go to the energetics and the analysis, you find that right side interaction is highly energetically favorable. So if the intermediate structure of different proteins are populated, there is a it's highly favored. The aggregation is highly favored or energetically favored. And they will lead to formation of a heteroprotein amyloid structure. And people have studied that heteroprotein amyloid structures are more toxic. That is why this process of amyloid aggregation is very complicated, and we are just beginning to understand this process. However, whatever the understanding we have, we can design some compounds to protect that and prevent that. So if you are interested, you can look at some papers, And the story is that we are talking about protein amyloid aggregation, protein prion particle aggregation, and now people are doing a research on single molecule self-assembly. You take tryptophan, you take phenylalanine, you incubate that, they will also form fibers. And these fibers have amyloid-like characteristic properties. So maybe in future research, amyloid aggregation is not limited to proteins. Maybe RNA or fragment of DNA, they can also form amyloid-like structures. So all these studies are there, and we are working on that. This is a serotonin. Have you heard about serotonin? It's a single molecule aromatic. If you put serotonin, they self-assemble and they form fibrous structures. And this is the simulated structure. All are self-assembly of serotonin at different nanoscale. And if you add this serotonin to the cells, they are going inside the cytosol and they are causing cell death. So serotonin, even though it's not a protein, can form amyloid-like aggregate structure, and these structures are also doing amyloid-like cytotoxicity. So this is one area. Just to visualize, look, these white molecules are serotonin, and these molecules are coming together and forming some oligomers, and they lead to formation of fibers. So it's not only protein which aggregates, single molecules also aggregate, and this aggregation is also leading to formation of cytotoxic structures, and they are important molecules. Serotonin is the molecule of happiness. So we have to explore that. You can see the right side, दिख रहा है? नहीं दिख रहा है. ये light off हो जाएगा. ये हुआ, ठीक है. You just focus on that circle. You see some red dots, and red dot is disappearing as a function of time, right? This is a live cell imaging. Red dots are aggregates, and these aggregates are going inside the cell. So the amyloid aggregates can go inside the cell, and this is the visualization of that. And once they go to the cytosol, then they do their cytotoxication. And this is the 3D image. Next thing, the concluding part, is the other way. You have amyloid aggregate from protein peptide to amyloid aggregate, and we also talked about single molecule forming amyloid-like structures. Now, crosstalk, we talked that one protein amyloid can trigger aggregation of other protein. Now, crosstalk between metabolites. So, the serotonin aggregates or phenylalanine aggregates can also trigger aggregation of proteins. There dilutes the relevance of prion. Prion was hype because infectious protein particles. But if serotonin aggregates can trigger aggregation of other proteins, then it also becomes an infectious particle. So it is getting diluted, that discovery. But what is the mechanism? If you see the picture from initial side, this is the beta strands, amyloid structures, and then single molecules can be trapped in the beta strands. And once beta strand traps single molecules, there are two factors. One is the molecule itself aggregates, we understand their intrinsic tendency. Now it got a platform, and the platform they are trapped. So the local concentration of the molecule increases, and concentration is a factor for triggering aggregation. So due to this, the amyloid structures of protein, I repeat, amyloid structures made up of proteins can trigger aggregation of single molecules. Similarly, the other way, amyloid aggregates or amyloid-like structures of single molecules can also trigger aggregation of proteins. Both the way it works, so it complicates the system, and that is what I want to show here. So cross beta structure of protein fibers can trap single molecules and induce their aggregation. So बीच में जो cross beta structure of protein का है, and these blue molecules are phenylalanine, the metabolites which are aggregated. So it traps and then trigger their aggregation. Okay, so I want to conclude, and this is my lab, and I am fortunate to have done postdoc in two important labs. One is Ronald Wessel, who is expert in Huntington's disease, and Barbara Brodsky. She works on collagen. You can see her name in the white and white. Collagen structure was discovered by her. Barbara Brodsky and Helen Berman, triple helix structure. And my PhD is from IIT Bombay. And these are my gang, amyloid gang. And this is the grants which I got. for the research, because without money you cannot do research. So if I want to summarize, I want to say that you have a protein which is forming amyloid fibers. You prevent that amyloid aggregation, and you also inhibit that amyloid fiber formation. At the same time, you also try to dissolve it. So hopefully, nano-based strategies can give us direction for the therapeutic development of amyloid. Not only we are working on amyloids, people are working on other diseases. So the message is most of the compounds that this piperine, which is showing in vivo, the best result that from our lab research is piperine, which is a spices, and we are working on capsaicin, myricetin, quercetin, lot of compounds. So I would say this: all of you will have lunch, right? So who knows when we eat spicy Indian curry? When I say spicy means piperine, capsaicin, all flavonoids, and then formulation. And we have to discuss milk one, right? Full of natural compounds and their formulations, we may be protecting ourselves from the devastating neurodegenerative disease. ज़्यादा spicy सब्जी खाएंगे शायद हमको Alzheimer disease ना हो. So I hope you understand, and I somehow gave some information about amyloid aggregation. My aim was to give the fundamental information. And the question of any question I told to discuss in the end. Milk. The milk is that curcumin is a molecule, and curcumin does not go to the solution very easily. It cannot dissolve in water, so it needs in. That is one thing. Second thing is the bigger question is milk is actually very huge composition, heterogeneous composition. Milk has proteins, milk has lipids. Have you if you heat the milk and you will see lot of droplets, but those droplets are actually vesicles. So curcumin is the active compound of haldi, and if you take curcumin, pure curcumin, and just keep it exposed, it will get oxidized. Like you cut the apple, the white color becomes brown in 15 minutes. Why? Because the flavonoids gets oxidized, and oxidized curcumin is not curcumin. So we need curcumin for our body system. So curcumin is good. So curcumin's bioavailability is a big question. In search, you'll find curcumin's bioavailability in the body system is a problem. If you add milk, and then these vesicles are there, being a hydrophobic curcumin molecule, it gets into, if there is a vesicle that is water, and if you have a hydrophobic molecule, then it gets into the vesicle because it goes down, and then it gets stabilized and get absorbed by the intestine and go to the bloodstream, and there is no issue of oxidation, so it does its function. So that is one question. Any other question? So I was in my hobby was doing cartoon, and I always try to represent my research in cartoon so that I convey what exactly we are doing to just inspire the youngsters, because they don't get into details, no. So this is amyloid venous. As I showed, 120 years have passed. We still have not been able to treat Alzheimer's disease. किसी का memory loss Alzheimer's disease होगा तो he has to suffer with a memory loss life. It's very tragic. And this is the inhibitor. Like we have strong compounds: curcumin, quercetin, flavonoids, eugenol, piperine. They are very nice, brave soldiers, but they are not capable of doing this. So there comes the nano strategy, and I call it like transformer. Have you seen transformer? Okay. So transformer maybe, and then it goes back, and then this maintenance is scary, and it may win that. So this is the gist of the story, is that even though you have very important natural compound or drug molecule, you have to give it to the system in a very good formulation so that its efficacy is very much enhanced. This is my thank you slide to all of you. My have you seen this bird? This is called bulbul bird, and this is a nest. In my opinion, this is one of the best housing architecture in birds. And my question is, which architecture institute, MIT, Harvard, or IIT, did this bird go to get the AC, air conditioned, earthquake proof, rain proof? Which institute? Where did it get the training to build such a beautiful architecture, hanging architecture, earthquake proof, rain proof? It's not a direct question, but I am asking this question for just inspiring. I want to inspire myself. It's the nature. Nature is the ultimate source for inspiration, particularly in science. Definitely, it inspires others also, music and lot of others. But for science also, nature is a very good source of inspiration. Three things. This bird knows what are the building blocks, the small twigs it collects. This bird also knows how to connect them. It does it because it weaves its structure. This bird also knows what is the structure that it is building. That architecture is in the bird's brain. These three things are very important for us also. Whether we are making a nice architecture building, we should have building blocks, bricks, we should have a structure, and we should know what structure we are building. Same thing applies for nanostructure or nano architecture. What we are targeting, what building block we are using to make that nanostructure, and what is the efficacy or ultimate structure that we are making. So let us take inspiration from nature for the building blocks, for the architecture design, and for the application time. Thank you very much. If you have any question, you can ask me, and by email, or you are here for 1 month, right? Anytime. If you have any questions, please ask me. This is a very important question. I don't have right answer because the moment I went into nano, I had also same gold nanoparticle, platinum nanoparticles. One is these are the very important metals which has been maybe practices. People, our Ayurveda also says Loha Bhasma, Swarna Bhasma. So those are also metals. So depending on the metal and also availability. One thing I would say that from my experience and the knowledge that gold nanoparticles are less toxic as compared to silver nanoparticles. And it is easily possible for making some coating, or particularly making nanoparticles coated with natural compounds. You have lot of flexibility when you are starting with gold nanoparticles. You know how to make gold nanoparticle? You don't start with bulk gold. You start with auric chloride, which is a salt solution, and it's like a reduction method. Au plus 4 to metallic Au0. So that reduction method is a reaction, and you have to do that reaction, add your compound capping agent, so that end of the reaction you get nanoparticles. This process is also very, very established in gold. And the main thing I would say it is biocompatibility. FDA has approved gold nanoparticle less than 5 nanometer, so it doesn't have any problem. So people are making that. Maybe future lot of other, any other. और कोई भी क्वेश्चन है आपके दिमाग में, यू कैन नॉट आस्क राइट नाउ, यू कैन आस्क मी एनी टाइम. आई एम इन स्कूल ऑफ लाइफ साइंसेस एंड यू कैन अप्रोच मी एनी टाइम एंड ईमेल मी. और ठीक है, एनी अदर क्वेश्चन? मैं एक क्वेश्चन पूछूं? Any compound. I told we have done many natural compounds. Piperine, the Parkinson's disease reversal is by piperine coated. If you have a compound you want to make a nanoparticle, you have to do the reduction reaction with that compound. If that compound, some natural compounds act as a reducing agent, so you don't have to add that capping agent. So they facilitate that reaction, Au plus 4 to Au0 reduction reaction. They facilitate that, At the same time, they get capped. That was the question. Question क्या था? Any amino acid. Our aim was to target hydrophobic. That is why you chose. Huh? We have used these three because we want to target amyloid aggregation. You can make any. People have designed nanoparticles coated. All amino acids can be coated with for different function. Like we made for collagen fiber formation. That is other research part I did not discuss. I discussed amyloid here. We made a glycine coated nanoparticle. Glycine coated glycine, hydroxyproline, and proline are the collagen sequence. So to prevent collagen fiber formation, we made hydroxyproline nanoparticles, gold nanoparticles. So you can make depending on your requirement. Okay. Anything else?